So in today's lecture, I'll discuss on purine synthesis or the de novo synthesis of purine nucleotides. So these purines and pyrimidines are the nucleotides which are required for synthesis of nucleic acid, for synthesis of DNA and RNA, and also for various other cellular functions. So we can see here like dietary nucleic acids, they are the non-essential component. Why they are non-essential? They have lesser use. They are least it's recycled in the body because this is the metabolic fate after being digested. Like dietary nucleic acid, DNAs and RNAs, they converts up into the mononucleotide under the influence of enzyme ribonuclease and deoxyribonuclease. These mononucleotides can further be converted up into the nucleoside in the presence of concerned enzymes with the loss of phosphate group. That further hydrolysis of these nucleoside lead to the bases and deoxyriboses as a separate entity. Further, the bases can be oxidized to carbon dioxide and ammonia, which can subsequently be excreted in the urine. So this is the fate of bases. Further, these ribose and deoxyribose sugars, these also can be absorbed in the body and they come into the blood. So this is the metabolic fate of these dietary nucleic acids. So for synthesis of DNAs and RNA, we have to be dependent on endogenous synthesis of these purines and pyrimidines. So here we'll see the biosynthetic pathway of purine nucleotide. In de novo pathway, why we are calling it a de novo? Because in this particular pathway, the purine ring is assembled on the ribose 5-phosphate. So ribose 5-phosphate will act as a platform for synthesis of purine. And the two parent purines are AMP and GMP. Okay? Adenine and guanine, and their concerned nucleotide AMP and GMP. So purine nucleotide can be synthesized by two pathways. We'll see here the first de novo pathway and then salvage pathway. So de novo biosynthesis of purine nucleotide. So we can see the structure of this purine base. So this base, actually this consists of two rings, and how we will name them? Two nitrogens are present in each ring. So we will number from 1, 2, 3, 4, 5, 6, and then 7, 8, 9. So what are the different components of the ring which are being contributed by different kind of substances? Like glycine is contributing C4, C5, and N7. So C4, C5, and N7 part of this skeleton is being contributed by glycine. Similarly, aspartate is contributing to nitrogen 1. Then glutamine is contributing to N3 and N9. So this N3, this is the N3 and N9. These are being contributed by glutamine. Then tetrahydrofolate is it's giving C2 and C8. So this is C2 and C8 are being derived from N10 formyl and THF. Here N5 and 10 methynyl and THF, right? And carbon dioxide is giving the carbon 6. So this is how the various components they are going to contribute to the various elemental skeleton of the this ring. Now we will see it at the formation. First, we will see during the course of synthesis, de novo synthesis of purine, there is first synthesis of PRPP, that is phosphoribosyl pyrophosphate, because on this platform there will be synthesis of purine. Purine cannot be synthesized as an independent entity. So the PRPP is being synthesized from glucose 6-phosphate, and via pentose phosphate pathway it is going to deliver ribose 5-phosphate. Because a championship or PPP pathway is important source of this ribose phosphate as well as NADPH. So this ribose phosphate is converted into the PRPP, and which can further be utilized for synthesis of purine nucleotide as well as for the pyrimidine nucleotide. So we'll discuss the purine nucleotide synthesis here. So ribose 5-phosphate. In the first step, there are total 11 steps in the synthesis of purine nucleotide. So we will see the steps like ribose 5-phosphate first converted up into the phosphoribosyl pyrophosphate in the presence of enzyme PRPP synthetase. Synthetase, why we are calling? Because ATP is utilized here. It will be converted up into the AMP, and the one phosphate is being utilized here. And so the PRPP is now being synthesized, phosphoribosyl pyrophosphate. Further, the glutamine is now contributing its amine group. So enzyme is glutamine PRPP aminotransferase enzyme. This enzyme is lacking in the brain actually. But other sites it is abundantly present. So most of the cell can have their own biosynthesis of these purine and nucleotide, except for erythrocytes, where there is this enzyme is lacking, and the partial, synthesis in the brain. So the glutamine PRPP aminotransferase enzyme, it is converting this entity PRPP into the 5-phosphoribosyl amine, and this glutamine converting to glutamate. Further, in the next step, the 5-phosphoribosyl amine is converted up into the glycinamide ribonucleotide. So the enzyme is glycinamide ribonucleotide synthetase. Here also 1 ATP is utilized. Further, in the next step, the glycinamide ribonucleotide formyl transferase enzyme is there. So formyl group is to be added, and the source for formyl group is N5-ion-10 methynyl tetrahydrofolate. So this is donating the formyl group, and this is going to add on to this glycinamide ribonucleotide. So in the step 4, there is a synthesis of formyl glycinamide ribonucleotides, FGAR. Then further in the next 5th step, again glutamine is contributing one more amino group. The enzyme is amidotransferase. So there is a formation of formyl glycinamidine ribonucleotide. Further in the next 6th step, the enzyme is pharma-aminoglycinamide midine ribonucleotides cyclase. So this enzyme, what it does, it leads to closure of the ring. There will be formation of ring with the loss of water molecule and the formation of amino imidazole ribonucleotide. So the first ring is imidazole ring is being created. In the next step, the imidazole ribonucleotide carboxylase enzyme, in the presence of carbon dioxide, it converts this entity into the carboxy amino imidazole ribonucleotide. In the next step, step 8, the aspartate is going to add here, and in the presence of enzyme synthetase, again ATP is being utilized here. The formation of N-succinyl 5-amino imidazole 4-carboxymide ribonucleotide is being there. Then in step 9, there is a loss of fumarate. Step 9 and step 11, there is a splicing and there is no addition in these steps. So here in step 9, the fumarate is lost and the remaining entity is now amino imidazole carboxymide ribonucleotide. Then in the step 10, again there is a donation of formyl group via the N10 formyl THF, and now which convert up into the THF, and the enzyme is transformylase, and the product being formed is formyl imidazole carboxymide ribonucleotide. Further, the synthetase enzyme leads to the ring closure, and with the formation of ring, there is a synthesis of the very first purine nucleotide, that is the inosine monophosphate. IMP is being formed here. Then further, IMP is the mother purine. From this, there is further conversion to other nucleotide like AMP and GMP. So we'll see it here. IMP in the presence of aspartate, there is enzyme adenosyl succinate. It is the adenyl succinate synthetase. 1 GTP is utilized here and it is converted up into the adenosyl succinate. Then further in the step 13, the adenosyl succinate is converted up into the adenosine monophosphate with the loss of fumarate. The enzyme is adenylosuccinase. So this is how the synthesis of AMP. Further, the IMP dehydrogenase can convert this IMP into the xanthosine monophosphate, which can be converted up into the guanosine monophosphate. So this is how the synthesis of GMP, XMP, and AMP. So in this step, there is an addition of glutamine. So glutamine is acting as a source of amino group. So we have seen here what we will see it here, like for the synthesis of ATP, AMP, there is a utilization of GTP. And in this synthesis of GMP, there is a utilization of ATP nucleotide. So this kind of opposite use of these nucleotides. Then further, these mononucleotide can convert up into the concerned dinucleotides and trinucleotides, diphosphates and triphosphates. So this is nucleoside monophosphate like AMP or GTP, GMP or XMP. These all are converted up into in the presence of kinase enzyme. One ATP is used here and phosphate is added here and it converts up into the concerned diphosphate. So if it is GTP or XDP, so they can be formed. Further from GDP to GTP. So this is how there is a synthesis of from diphosphate, from monophosphate to diphosphate and further to triphosphate. So we will see in this slide what are the regulatory steps. So we have seen 11 huge volley of reactions are going on. So three important steps. The step 1, which is being channelized by PRPP synthetase enzyme, and that is the regulatory step. And the second is the glutamine PRPP amidotransferase, and third is the adenylosuccinate synthetase enzyme. And this is IMP dehydrogenase. All these enzymes are being regulated through feedback, negative feedback inhibition. So if there is sufficient amount of AMP or GMP, so through negative feedback inhibition pathway, they are inhibiting these enzymes. So that indicates there should not be any more synthesis because the AMP and GMP are in sufficient amount. So it suppresses the synthesis or expression of these proteins. So these are the three important enzymes, regulatory enzymes. Out of these, this glutamine PRPP amidotransferase is the rate limiting enzyme as well. And this PRPP synthetase mainly being regulated by ADP and GDP. And this PRPP amidotransferase enzyme is negatively inhibited by IMP, AMP, and GMP. So this was all about the de novo synthesis of purine nucleotides. Another pathway of synthesis of purine is through salvage pathway. Salvage pathway is a very important pathway because this involved the direct conversion of purines to nucleotide. What we have seen earlier in the de novo synthesis, initially there is a PRPP is being required. And on that platform, there is a direct synthesis of nucleotide. Not the independent purines are being formed first. But here, the direct addition of few purines to PRPP, and that leads to the synthesis of these nucleotides. So this pathway is known as salvage pathway. It consists of single reaction. So few purine bases like adenine, guanine, or hypoxanthine, which can be formed in the cell during the metabolic degradations of the nucleic acid or nucleotides, which can be salvaged and used over and over again to remake the purine nucleotides. So this is the best use of through recycling of these purine and pyrimidines. So we will see this reaction, Solvay's reaction by phosphoribosylation of adenine, hypoxanthine, and guanine. So adenine is going to add to PRPP, phosphoribosyl pyrophosphate, And the enzyme is adenine phosphoribosyl transferase, because adenine is converting to AMP, so it is adenine. So there is a direct formation of AMP. It's single step conversion. This is the beauty of this salvage pathway. Similarly, hypoxanthine to inosine monophosphate. And here the enzyme is hypoxanthine guanine phosphoribosyl transferase. Similarly, guanine can also be converted to GMP via the same enzyme, that is the hypoxanthine guanine phosphoribosyl transferase, right? And this excess amount of AMP and GMP will regulate these enzymes through negative feedback regulation. Then salvage reaction by phosphorylation of purine nucleotides. So there is another way by which the purine nucleotides can be directly synthesized, means the direct addition of ATP onto the purine nucleoside. So purine nucleoside are being added with the phosphate group donated by the ATP in the presence of kinases, and that converts nucleoside now converted to nucleotide. So this is also a type of salvage reaction, single step reaction. Now, what is the significance of salvage pathway? Because we know that HGPRTase channels the hypoxanthines and guanine back into the DNA synthesis. We have seen this in the single step. These can be used for synthesis of these hypoxanthine and guanine back into the DNA via the salvage pathway. If there is a failure of this enzyme, what will be the consequence? So the first there will be, excess amount of these purines. Purines will be in the excess. So these purine are now degrading to uric acid. The first drawback is then increased concentration of uric acid, which is a major purine breakdown product. Then second is the de novo pathway will be stimulated, because now the PRPP is in excess quantity. So that de novo pathway has to be, it is to be stimulated manifold. Then second important aspect is there, like salvage pathway provides the purine nucleotides for tissues which are incapable of biosynthesizing via the de novo pathway. As I've already explained, like the brain, there is a low level of PRPP, aminotransferase. So therefore, it is partly dependent on de novo pathway as well as salvage pathway. So here comes the importance of salvage pathway. Secondarily, erythrocytes and polymorphonuclear leukocytes, they cannot synthesize 5-phosphoribosylamine, and therefore utilize the exogenous purines to form the nucleotide via a salvage pathway. So in these two important tissues, there is a special importance of salvage pathway. Then we will see here the degradation of purines. The ultimate degradation product of these purine nucleotide is always the uric acid. So you can see the fate AMP via the nucleotidase converts up into the adenosine with the loss of phosphate group. Then in the presence of enzyme ADA, adenosine deaminase, this enzyme, which converts adenosine to inosine, and this ionosine in the presence of enzyme nucleoside phosphorylase, it converts up into the hypoxanthine. So this is how there is a conversion to hypoxanthine. Similarly, GMP can be converted up into the guanine via guanosine nucleoside formation. Then further, we can see it here, this hypoxanthine and guanine via the enzyme guanine oxidase and guanase. These two enzymes convert these two entities into the xanthine itself. So ultimately, whether it is hypoxanthine or guanine or adenine, all of these finally convert up into xanthine. This xanthine is finally converted up into the uric acid via the same enzyme xanthine oxidase. So this xanthine oxidase utilize the water which converts up into the H2O2. Now this uric acid is generally excreted through urine, and the absence or like in patients suffering from gout, there is a special importance of this enzyme. We use some competitive inhibitor against this xanthine oxidase which arrest the activity or which competitively inhibit the xanthine oxidase activity. So we can make use of allopurinol in order to decrease the concentration of uric acid. In the patient suffering from gout, in the patient suffering from von Gehrig's disease, and the patient suffering from Lesch-Nyhan disease, because there is a secondary manifestation in these two diseases is increased level of uric acid, right? In Lesch-Nyhan disease and the von Gehrig's disease. Then here is the importance of adenosine deaminase. So in the absence of this enzyme, there is a disease SCID. So we'll discuss further about these gout and SCID in detail in further lectures. Thank you.